Hello and welcome to the vodcast about Year 9 homeostasis. We're going to start with the control of the body, and in particular the nervous system. Above the word nervous system, you'll see a blue neuron that we'll talk about now. The cell body, otherwise known as the soma, has dendrites that detect a stimulus, which is either a neurotransmitter or the environment. The axon is the arm that travels from the soma to the axon terminals. The axon can have a myelin sheath made by Schwann cells. If it does, we say it's myelinated, and the myelin sheath increases the conduction velocity of the neuron, and the conduction velocity is the speed with which the message travels along the axon. The axon terminals release neurotransmitter into the synapse. The synapse is the gap between the neuron and the cell that it is messaging. This is either the effector or another neuron. The message delivered by neurons is directly to the site of action, whether this is the effector, another neuron, or muscles or glands. We say that the nerve message is electrochemical, electro because of the action potential, and chemical because of the release of neurotransmitter. Upon an action potential arriving at the axon terminal, the neurotransmitter is released into the synapse, which it then crosses and then attaches to a receptor on the effector cell. Neurons are extremely fast. Even unmyelinated neurons have an action potential speed of 2 meters per second, but the myelinated, which means it has Schwann cells wrapped around the axon, is 140 meters per second. The neurotransmitters are only short-acting. Once it hits the receptor on the effector cell, it's rapidly broken down. There are 3 types of neurons: motor neurons, interneurons, and sensory neurons, and there's a picture here below of the 3 types. Let's now move on to the endocrine system. Endocrine glands are ductless, which means they have no duct, unlike exocrine, like a sweat gland, that does have a duct. Endocrine glands release hormones directly into the bloodstream. The hormone will travel around the entire body in the bloodstream until it reaches a cell with the correct receptor that we call the target tissue. Hormones alter metabolism. Hormones take longer to take effect, and the duration of action is also longer than that of a nerve. Hormones of note are thyroxine, released by the thyroid gland, adrenaline, released by the adrenal medulla, which sits just above the kidney, insulin and glucagon, that are released by the pancreas, antidiuretic hormone, that is released by the posterior pituitary just underneath the brain, and testosterone and estrogen, that are released by the respective gonads. Next, we're going to talk about the reflex arc. The reflex arc is a protective mechanism. It's automatic and subconscious. The receptor detects the stimulus. The sensory neuron takes the message to the spinal cord through the dorsal root ganglion. The sensory neuron synapses directly with an interneuron, and the interneuron synapses directly with a motor neuron. The motor neuron takes the message out to the effector through the ventral root. The effector carries out the response without the brain even knowing. The brain will find out later on. And once again, the purpose of the reflex arc is to remove the person from danger. And here I've got a simplified diagram of the reflex arc, and then a more detailed diagram showing that the receptor here is detecting pain, and then a sensory neuron is coming into the spinal cord through the dorsal root ganglion, where it synapses with an interneuron, that synapses with a motor neuron. The motor neuron then exits through the ventral root, goes to the effector, and the response is to withdraw the hand from the painful stimulus. Next, we're going to talk about cellular respiration. Cellular respiration is an important factor when considering thermoregulation, which we'll get to in just a minute. Cellular respiration occurs in the cytoplasm and mitochondria of cells. Glycolysis is the first step, and it occurs in the cytoplasm. Glucose is converted into 2 pyruvate molecules. This is an anaerobic process, which means it doesn't require oxygen, and it makes 2 ATP. If no oxygen is available, then the pyruvate stays in the cytoplasm, and it's used in anaerobic respiration. The products of anaerobic respiration depend upon what cell type you are. Plant cells make carbon dioxide and ethanol, whilst animal cells make lactic acid. Now, if oxygen does become available, the pyruvate is shunted into the mitochondria. A shunt is just an active process moving the pyruvate from the cytoplasm into the mitochondria. Pyruvate then enters the Krebs cycle, which is inside the fluid matrix of the mitochondria. The Krebs cycle makes 2 ATP. ATP is the energy currency of a cell. By adding an inorganic phosphate, which is Pi, to ADP, we end up with ATP. The phosphate bond is the store of energy. Next comes the electron transport chain that occurs in the cristae of the mitochondria. The cristae is the internal folded membrane of the mitochondria. The cristae is folded to increase the surface area to volume ratio, which increases its efficiency. The cristae is studded with ATPase. The proper name for ATPase is ATP synthase, which makes ATP by joining an inorganic phosphate to adenosine diphosphate to make adenosine triphosphate. The electron transport chain makes 34 ATP and also releases carbon dioxide and water. On the whole, the substrates for cellular respiration are glucose from the digestive system, oxygen from the respiratory system that then travels through the cardiovascular system, and the products are carbon dioxide that exits through the respiratory system, water that exits through the kidneys, lots of ATP in the presence of oxygen, and heat that heats our body. And here I've got a diagram that shows you both glycolysis, the Krebs cycle, and the electron transport chain in relation to where they occur. Glycolysis occurs in the cytoplasm, whilst the Krebs cycle and the electron transport chain occur inside the mitochondrion. Okay, another thing we need to consider is the effect of radiation. Radiation is the transmission of energy in electromagnetic waves. There are two kinds of radiation, ionizing and non-ionizing. Ionizing radiation is high-energy radiation that is able to remove the electrons from atoms, which is why it's called ionizing, because it creates ions. Examples of ionizing radiation are X-rays and gamma rays. They can cause mutations in the DNA that can lead to cancer. Non-ionizing is low energy that can only cause heating. Microwaves, UV, and infrared are examples of non-ionizing radiation. They can burn the skin and damage the eyes. Okay, now we get onto the homeostasis part. Homeostasis is the maintenance of the internal environment despite changes in the external environment. It uses the stimulus response model, and here's a little diagram I've made of the stimulus response model. The stimulus is the change in the environment. The receptor is a modified dendrite that detects the stimulus. The modulator decides upon the appropriate response. The effector brings about the response, and the response is the overall desired reaction to the stimulus. When the response opposes the original stimulus, we call it negative feedback. And when the response increases the original stimulus, we call that positive feedback. Okay, now we're going to look at thermoregulation, which is how the body regulates temperature. So we'll look at what happens if the body gets too hot. The stimulus is an increased blood temperature. The receptor is a thermoreceptor in the hypothalamus. The modulator is also the hypothalamus, in particular the thermoregulatory center. Neural messages are sent from the modulator to the effectors, and here the effectors are sweat glands, blood vessels, and erector pili. Erector pili are the little muscles at the base of every hair. The sweat glands produce sweat. The blood vessels vasodilate, which means the blood vessels come to the surface of the skin, and the erector pili make the hairs relax. All of this allows the heat from the blood to heat up the water in the sweat, and when it evaporates, it takes the heat with it, and this is called evaporative cooling. The response then is a decreased blood temperature. And this is negative feedback because the response opposes the original stimulus. But what if it's too cold? The stimulus is decreased blood temperature. The receptor again is a thermoreceptor in the hypothalamus. The modulator again is the hypothalamus, in particular the thermoregulatory center. But this time the messages from the modulator to the effectors are both neural and hormonal. In particular, thyroxine is released from the thyroid gland. And the effectors are skeletal muscle, blood vessels, erector pili, and the liver and somatic cells. A somatic cell is a body cell. The skeletal muscle contracts at 20 Hz, and we call this shivering. The blood vessels constrict in a process called vasoconstriction that takes the blood vessel away from the surface of the skin. The erector pili cause the hairs to stand on end, and we call this piloerection. You might call it goosebumps. And the liver and somatic cells increase the rate of cellular respiration to generate heat. The thermogenesis from cellular respiration and shivering and the prevention of heat loss through goosebumps and vasoconstriction allow us to reach the response, which is an increased body temperature. This is also negative feedback because the response opposes the original stimulus. Next, we're going to look at glucose homeostasis. So, we're going to look at what happens if you eat a meal. The stimulus here is increased blood sugar. The receptor is a chemoreceptor in the pancreas, in particular the beta cells. The modulator is the pancreas, and in particular the islets of Langerhans. And it causes the release of insulin. The insulin has effects on both the liver and the somatic cells, which are the effectors. The liver undergoes glycogenesis, which is the creation of glycogen, and lipogenesis, which is the creation of fat. The liver also increases its rate of cellular respiration, generating more heat and more ATP. And somatic cells also increase their rate of cellular respiration, generating more heat and more ATP. The effect of all of this is to store the excess glucose and use it to generate ATP. Therefore, the response is decreased blood glucose because glucose has come out of the blood and into the effectors. This is an example of negative feedback because the response opposes the original stimulus. Next, we'll look at fasting, or you haven't eaten for a while. The stimulus is decreased blood sugar. The receptor is the chemoreceptor in the pancreas, but this time it's the alpha cells. The modulator is the pancreas, and in particular the islets of Langerhans, and this causes the release of glucagon from those alpha cells. The effector is the liver that undergoes glycogenolysis and lipolysis, so it's breaking down glycogen back into glucose and breaking down fat into its precursors that can be used in gluconeogenesis. Gluconeogenesis means using something else other than glucose to drive cellular respiration. All these mean that glucose is released from storage and other fuel sources are used for cellular respiration. The response here is an increased blood glucose level. And again, it's an example of negative feedback because the response opposes the original stimulus. Now we'll look at water homeostasis. So, what happens if you haven't drunk enough water? Well, you become dehydrated. The stimulus is decreased water in the blood. Another way of thinking of it is that you've got too much salt in the blood, and salt is a solute inside the blood. The receptor is an osmoreceptor in the hypothalamus. The modulator is the hypothalamus, and this causes the release of antidiuretic hormone from the posterior pituitary, which sits just below the brain. The effectors here are the distal convoluted tubule, which inserts water pores into the nephron. The collecting duct, again, inserts water pores into the nephron. An aquaporin is the proper name for the water pores. And it also initiates the thirst reflex that makes us drink more water. The effect of this is scant, concentrated urine, which means that your urine comes out in a small volume and quite dark in colour, and also you want to drink water. So the response is an increased amount of water in the blood. Again, this is negative feedback because the response opposes the original stimulus. But what happens if you drink too much water? The stimulus is too much water in the blood, or not enough salt in the blood. The receptor is the osmoreceptor in the hypothalamus. The modulator is the hypothalamus, and this causes a reduction in the release of antidiuretic hormone from the posterior pituitary. The effectors are the nephrons, which reabsorb less water, and the thirst reflex is switched off, and you don't feel like drinking water. The effect of this is copious dilute urine, so you're urinating a lot and it's very light in colour, and you don't feel like drinking water. Therefore, the response is decreased water in the blood, which raises the amount of solute in the blood comparatively. And again, this is an example of negative feedback because the response opposes the original stimulus. Now we get to the last part of my mind map, and we're looking at immunity. So the immune system maintains a stable internal environment and makes sure that the only cells inside your body are your own. The immune system defends against pathogens. And there are 3 lines of defense. The first line of defense prevents entry. It's non-specific, which means it doesn't care who you are, you're not coming in. And the examples are cerumen, which you might know as earwax, that traps pathogens, tears that flush away pathogens, stomach acid that kills pathogens, and the skin is a physical barrier that prevents pathogens gaining entry. Mucus traps pathogens, and the ciliated epithelium moves the mucus from the respiratory tract into the stomach for destruction. Once the pathogen bypasses the first line of defense, normally through a lesion, which is a cut, it will encounter the second line of defense. And once again, it's non-specific because it will attack anything that doesn't belong. And the main cell type here is the macrophage, and it uses phagocytosis to engulf pathogens. And when it engulfs the pathogens, it creates pus. The macrophage can also initiate fever and can also initiate the third line of defense. Now, the third line of defense needs the second line to be activated to become functional. The third line of defense attacks specific pathogens, so we say that it is specific. There are 2 types, being humoral or cell-mediated. Sometimes humoral is called antibody-mediated. Both create memory cells which are important for a rapid response in case of a second infection. Humoral immunity creates antibodies, and the antibodies attach to the pathogens, which leads to the ultimate demise of the pathogen. And cell-mediated kills infected cells, and it kills the infected cells using cytotoxins, which is a toxin that kills cells. Sometimes this is called induced apoptosis because it makes the cells self-destruct. And here we have a diagram that shows you that the pathogen first encounters the first line of defense, and if it bypasses this first line of defense, it can be attacked by a macrophage. And the macrophage causes inflammation and destroys the pathogen through phagocytosis. But the macrophage is needed to initiate the third line of defense. And we see here for antibody-mediated, which is humoral-mediated, that B cells create plasma cells, which create antibodies. It also initiates the creation of memory B cells, which are needed in case of a second exposure. And that it also initiates cell-mediated immunity, and cell-mediated immunity creates cytotoxic T cells, which destroy infected cells, and it also creates memory T cells in case of the second exposure. Thank you for paying attention. I hope you've liked it. Don't forget to like and subscribe if you've made it this far, and I'll catch you later. Bye now.